In Chapter 8, we'll investigate the genetics of cancer. And this chapter is going to be a bit longer than the last one because cancer is actually a disease of the DNA. And some of our newer techniques with sequencing and everything like that is being used now with determining the aggressivenesses of cancer and then basing that on treatment and other things. So cancer is at the forefront for a lot of these genetic techniques that we're talking about and how they're going to be applied. So let's get into it. Oh, and this lovely picture that we have here on the slide is colon cancer, obviously a very obstructive one, which you can see right here. Let's start out with a few facts about cancer, which I am sure you are all well aware. 1 in 4 deaths occur to cancer. More than 50% of them are diagnosed when the cancer is already invasive. And of course, the cancer frequency increases as you age or the population ages. Cancer is a genetic disorder, but it isn't necessarily inherited. Inherited cancers are actually rarer, and these, of course, are mutations that are present in the chromosomes in the germ cells. So the child inherits an increased risk to develop cancer. Most cancers are a result of accumulated exposures to environmental agents that induce mutations in the DNA. And that's why the longer you live, the more likely you have a chance to be exposed to something that eventually led a cell to become immortal, is how we have a tendency to refer to it. Now, we do now have what we call the Cancer Genome Atlas. And this is a creative catalog of all the different genomic changes that have been reported in multiple types of cancers. And of course, it's at the NIH. So if you want to take a look at it, and whenever I get in there, I again get lost because I did some work on cancer. It was at the NIH at the Cancer Institute for 5 years. So yeah, this is where I find things very fascinating. But anyway, so it's very easy to get into. And once you get familiar with how the website works, you can get kind of engrossed rather quickly. Well, what is cancer? It's a disease of DNA, and that leads to uncontrolled cell growth. Now, most of the time, cells are listening to the environment. They're responding to signals other cells are giving it and everything, but cancers aren't. And that's why we call it uncontrolled cell growth and why we refer to them as immortal. Because most cells can only divide a certain number of times and then they die. That's not true for tumor cells. Anyway, key events required for the formation of tumors, tumorigenesis, include resistance to growth inhibitory signals. In other words, when there are enough cells around, the cells should be receiving signals that says, do not divide, do not divide. And if they don't, they keep dividing. Another way is all cells have within them sort of a suicide pathway, if you want, apoptosis. If that pathway gets inactivated, then they can stay cycling and continue to grow. Induction of angiogenesis. If they're growing and dividing actively, they need a lot of oxygen and other nutrients that are in the blood supply. So they produce angiogenic factors, which increase, actually, I should say not increase, but start the formation of new blood vessels. And then they have to acquire the ability to invade further into a tissue and then to spread to other tissues. So invasion and metastasis. And all of these, in many ways, are the accumulation of more and more mutations that inactivate one pathway, then another, and so forth. So, however, it's important to remember that all tumors originally arise from a single cell. So initially they're all monoclonal. Remember, as they divide, they're completely copying each other. So if nothing happens from that first mutation, when they're able to now divide indefinitely, if you will, they should all be identical. But remember, mutations occur as they divide, so they can accumulate, and different strains of tumors then have different powers. And this is what we see with someone with advanced cancer. I think most of you are familiar with what we call the traits of benign versus malignant tumors. Malignant being, of course, life-threatening, benign being compatible with life, unless it's impacting on something that could eventually, make herniate the skull or the brain and this sort of thing. So then we got to get rid of it. Malignant typically grows faster than benign, but not always the case. Definitely, malignant is able to invade local tissue and spread or metastasize to distant tissues. Benign, they're nice and easy to remove because they stay localized and they have a capsule around it, so when the surgeon comes in, they can pop it out and you don't feel like you're breaking it open and releasing cells. And then malignant, if you recall that first picture on the first slide, malignant tumors do not look like the tissue from which they arose. So they look like something entirely different, and that's what anaplastic is referring to. Whereas benign tumors still kind of look like the tissue from which they arose, and sometimes they continue to function. So let's see a better comparison with the next slide. And again, here is an example on the top of benign polyps. I've had a number of them. I think we all get a little bit, but you can still see the majority of the tissue here of the colon looks pretty normal. Now, however, with colon cancer, you can see this mass, which is a tumor, which is obstructing flow, obviously, doesn't look anything like these polyps or really any part. And look at the regular tissue where I'm showing with my little cursor there. That doesn't, it has a little bit of the top, but not much. So this is what this whole anaplastic is referring to. What are the causes of cancer? In this case, the genetic causes of cancer. Well, genetic alterations in genes that code for the proteins that regulate a cell's movement through the cell cycle. Now you know why, and I mentioned this in the previous lectures, of course, the importance of the cell cycle. And if those, especially the checkpoints and stuff, if they get inhibited, then the cell keeps moving through the cycle and starts to divide and make more copies of itself when it's not needed. So, you can induce it by inactivating or activating certain genes. And this is one thing we're going to really focus on. And I want you to try to keep these straight. So that's a big hint. And you can reverse the phenotype, which is ultimately what we would like to do, by adding back the normal genes, which would mean they would have to listen to, we don't need any more cells here right now, so stop growing, and they would. That's what normal tissue does. So wouldn't that be nice if we could do that? Well, that's what a lot of research is working toward. So, as I've already mentioned, genetic mutations occur during the lifetime of somatic cells. If you have too much sun exposure, many different things can lead to spontaneous mutation. And because, we're exposed throughout life, if you don't want to have a risk of cancer, then you don't want to be born. That's just the way it is. The frequency will increase by exposure to a carcinogen as you get older. And when they occur in the somatic cells, kidney cells or liver cells or something, we're not going to pass it on to our children. So it can't be inherited. Again, however, if it occurs in the germ cells, it can. But as I've already said in a previous slide, inherited forms of cancer are relatively uncommon. And here, of course, is a family tree with a history of colon cancer. This, if you take a look at this, again, of course, the darkened symbols represent individuals diagnosed with it. You can see that both males and females are affected, in which case you would have to say that this is autosomal as opposed to sex-linked. And you can see that this particular family had some real problems here. While our risk of developing cancer increases when we are exposed to chemicals and other environmental factors such as radiation, which just increases the frequency of mutations. Now, environmental agents, though, may not actually induce a mutation, but they can enhance the growth of cells that were altered without inducing new ones, and these are called promotion. So first of all, you're exposed to some sort of factor that will induce a mutation, and then another factor might promote its growth without inducing further mutations, and we call that promotion. And we'll talk about the various stages of cancer development in just a little bit. Now, known carcinogens and mutagens, in other words, which induce mutations, include, obviously, cigarette smoke. That's number one. Asbestos, coal, uranium dust. Again, the uranium is giving off radiation, ionizing radiation, which is why it's there. The UV light, and, of course, diet. One's response to those carcinogens, however, are going to be dependent on your genetic background. So even though you may be exposed to all of this, if you are lucky to have a particularly good DNA repair mechanisms and stuff, you might not get any cancer at all, even though you've been a heavy smoker for years. However, I wouldn't want to chance it. This is what I was talking to earlier in a previous slide, the multi-step theory of carcinogenesis. And this has been around for a long time because early on we realized that just a single mutation is not going to give a malignant cell all the characteristics that it needs to be fully malignant. So it requires more than one mutation or a multi-hit. So for many years we've said the steps of carcinogenesis are as follows. Now remember, anything like this is always, especially when it's a theory, is in development. As we understand more, this will be expanded or removed. But right now it kind of explains basically what's really happening. So the first step is known as initiation. A genetic mutation occurs so that the sequence of nucleotides in a chromosome has been altered. It's an irreversible event and it has an effect on a gene. Promotion, then, the mutant cell is induced to proliferate and pass on the mutation. If only one cell has this mutation and it never divides, it's not going to hurt you. Okay, but progression is where it begins to acquire different mutations and to exhibit full malignancy. And so there's going to be, it's going to take a while. So the older you get, the longer the time and the increased likelihood that a single cell that initially got a single mutation can now go on to invade other parts of your body. So, what are these genes that get mutated which lead to a cancer phenotype? Well, these are genes that normally control growth and differentiation in cells. So far, more than 100 of them have been identified. And they basically have turned out to be, again, proteins involved in cell division. We've identified how these processes become deregulated, which then allow us to understand how the process is regulated to begin with. So we find that there are three major classifications of cancer genes. Yes, I will expect you to know these. They are oncogenes, okay, cancer genes, oncology. These promote cell division. When they basically they are on all the time. When normally they're not supposed to be. They're only supposed to be on at certain times. That's when they function normally because they're responding controls. But an oncogene does not respond to controls anymore, and it just promotes cell division. A proto-oncogene, as you see here, is the normal cellular gene that promotes cell division under the right circumstances. But oncogenes, it doesn't matter what the environment is, it promotes proliferation or cell division under all circumstances. Then there are genes that we call tumor suppressor genes. And they basically are the things that stop the cell from going through the phases in the cell cycle. And if you lose those, okay, again, cell division will occur. And then, very importantly, there are DNA repair genes. And these DNA repair genes are very important. They code for proteins that will remove damaged sequences or damaged nucleotides and then repair things. But if one of our mechanisms or one of the proteins in there gets mutated and doesn't function properly, then things don't work. In this figure, we can kind of see what will happen to a normal somatic cell, a normal cell. So you'll get various environmental signals, such as hormones or cell-cell interactions. And if there are a lot of cells around and we don't need this cell, it might induce cell death. Okay, those would be the apoptosis or tumor suppressor genes. Okay, suppressor because they're killing the cell off so it doesn't get a chance to become a tumor. But in this case, the cell just isn't needed for the body, so we get rid of it. On the other hand, if the cell receives growth factors or steroid hormones that bind to its receptors, then it will grow and divide. These are the proto-oncogenes, and also usually for differentiation as well. So you can kind of think, well, this is what will happen. They will differentiate, grow, or die, and so the other genes, the cancer genes, are ones that are involved in a somatic cell's response to changes in the environment. Let's talk about each of these categories in a little bit more detail. So we'll start off with the proto-oncogenes, and this is where students have a little problem because proto-oncogenes just means that they're a normal gene. They have not sustained a mutation, and so they behave normally. Okay, so it's a gene present in the normal human genome, and it has a role in promoting cell growth or proliferation. Okay, in other words, it can go on and divide and make copies of itself. However, if a mutation occurs and now these genes are always on, even if a growth factor is not present, say, like a receptor, then we call it an oncogene. So an oncogene is a mutated proto-oncogene, and that's what you have to keep straight. So, as you see here, the proto-oncogene is a normal gene and promotes cell growth under the right conditions. Then it gets exposed to a carcinogen, and it becomes an oncogene, in which case it has a mutated gene and promotes growth, cell growth under almost all conditions, and that's not what we want. The four major functions of proto-oncogenes reflect how a cell responds to its environment and then begins to divide. So one proto-oncogene, a function of a proto-oncogene, is as a growth factor. Now these are small peptides. They're going to be secreted by cells, and then these growth factors bind to specific receptors on this target cell and induce growth. So if you have too much of a growth factor, if too much is being made, then you promote cell growth. Not surprisingly, then, and the next major function of a proto-oncogene is the growth factor receptor. So this is the receptor on the cell. It's on the membrane, so it will bind the growth factor, and it will then send a signal to the cell. Growth factors are here. We need to divide. Now, that going from the receptor binding to a change in cell behavior is that whole process of signal transduction. So anything in these pathways, enzymatic pathways and everything, that get activated so they're on all the time, and the signal transduction molecules gives the cell unregulated growth, uncontrolled growth. It's just growing, 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 and as you get enough of the cells, it forms a solid tumor. And then, of course, in order for the cell to divide, it has to produce all the enzymes, etc., that are needed to go through each of the phases of the cell cycle. And in order to do that, you have to have the transcription factors that promote the production of those proteins that induce cell growth. So not surprisingly, a major function of a proto-oncogene is nuclear transcription factors. So this is really showing you how cells respond to signals, growth promoting signals. These four major functions of the proto-oncogenes then are quite logical if you know how cells bind and then change their behavior. So let's look at the next slide to see it a little better. If you take a look at it, here's the growth factor, one of the major functions. Here's the receptor. Okay, then it will activate the signal transduction molecules, and if there are too much of them, then this activates transcription factors that will make proteins that help the cell signaling. And look at this, we're back to the central dogma. So we have the transcription into messenger RNA and translation into the protein. Now, oncogenes, okay, so these are the mutated proto-oncogenes, are dominant. Only a single copy of a mutated oncogene is required. You don't have to have two copies. And when they occur, it's a gain-of-function mutation. So it's too much growth factor production. It's, the receptor is telling the cell to divide, even though there's no growth factor there. It's an overactivity that results in uncontrolled cell growth. They rarely participate in inherited cancer syndromes. It just turned out to be that way. But they were first identified by studying viruses that induce cancer in chickens, and then by chromosomal translocations. We talked about that a little bit already, and what happens is this will take a proto-oncogene under its normal control. Now remember, a chromosomal translocation, we're exchanging between two non-homologous chromosomes. Oh, all those wonderful terms. All it means is, say, chromosome 2 is exchanging with chromosome 10. But the very first one that was identified was in chronic myelogenous leukemia, or CML. And this was chromosome 9 and 22, or what we call the Philadelphia chromosome. And what happened is it took this BCR gene, okay, and put it in a under different controls through that exchange with the chromosomes, so that now it became an oncogene that was always promoting growth. And this is how the CML arose, the chronic myelogenous leukemia. Of course, this was occurring in white blood cells. So how do oncogenes get activated? Well, they can get activated when a virus brings in an oncogene. In other words, what we usually call them is viral oncogenes, or V-oncs. And typically a virus will have picked it up from a previous host cell, but it's a mutated form of the proto-oncogene, so it's an oncogene. The virus brings it in, and it starts getting the cell to divide, which actually helps the virus replicate. And examples of viruses that do this are Epstein-Barr virus, HTLV1, human T-cell leukemia virus, and of course human papilloma virus that is associated with cervical cancer. Otherwise, you can get oncogenes from mutations in proto-oncogenes. Now, it can be within the gene, or think about it, you will have too much of something being produced, it would be in the regulatory sequence. So it could be in the promoter region, so RNA polymerase binds better. Chromosomal translocations will also do that, as we saw with the Philadelphia chromosomes. And sometimes, for whatever reason, the gene gets duplicated, so you have too much of it around and you just produce too much, so gene amplification. And this is exhibited in the figure on the next slide. So you see here, here's the normal gene product in the DNA, and you would get this protein. Gene amplifications, you have multiple copies of the gene, so you get too much protein. Point mutation, abnormal amount of a hyperactive protein, normal amount. And then chromosomal rearrangement, then you get, again, too much being produced. We don't have the virus in here, but it would be sort of the same thing. You have too much. Well, now, how do viruses induce transformation of normal cells? And this, I know you don't have this picture. I'm going to put it up in the unit anyway so that you can get it. But insertional immunogenesis, we've already learned that they can bring in oncogenes, but sometimes they can come in, and some viruses, not all, will insert into our chromosomes. And when they insert into our chromosomes, they can affect the production of that particular of the gene. And if it's a proto-oncogene, it might activate it or amplify it, as you see in these pictures. Now, sometimes the virus just induces other cells to proliferate in response to its presence, and those cells then sustain a mutation and they become cancer, not the cells directly that are infected. So, what are the oncogenic viruses? Well, the DNA viruses are the papilloma viruses, the cervical cancer, hepatitis B and C, liver cancer, and a lot of the herpes viruses, particularly Epstein-Barr virus with nasopharyngeal cancer. I had a student just this last semester who had a sister who got nasopharyngeal cancer after exposure to Epstein-Barr virus. Hodgkin's lymphoma, possibly. Oncogenic RNA viruses and retroviruses include the first one that was ever discovered, the Rous sarcoma virus, which actually causes an erythroleukemia in chickens, which doesn't sound all that serious unless you're a chicken farmer and it's wiping out your flock. Anyway, a virus can pick up a gene from a previous host cell and then infect a new cell and carry it in and then lead to transformation. And so here is a picture of the transforming retrovirus that I talked about, the Rous sarcoma virus, and it would come in with the oncogene, and then you've got a bird with a tumor. And you typically can't sell them very well after that. Now, how do we go about confirming the role of an oncogene? Well, we do this by something we call transfiction studies. I want you to think of them as addition experiments. We're going to add the gene to a normal cell and see if it becomes transformed. And we can do this in a number of ways. We can take normal cells and kind of inject the abnormal gene into it, or we can make a transgenic animal where a fertilized egg, we add the gene into the fertilized egg, and then put the fertilized egg into a mouse typically, and then you observe the offspring. If you're just doing it in the lab, then you're looking for the cells to clump on top of each other, and this is what you see typically if it does cause or induce transformation. So you're seeing some of the pictures here on this slide. Well, that leads us to a discussion of tumor suppressor genes. Remember, we have tumor suppressor genes and DNA repair genes to talk about. Now, we've identified so far more than 20 tumor suppressor genes, and they're kind of confusing. And here's why. You would think tumor suppressor genes should be a recessive mutation, because now it's a loss of function, because normally they stop the cell from cycling. And so if you inactivate one gene on the one chromosome, you still have a normal gene on the other chromosome. And so typically in a recessive situation, that would be fine. But the problem is the heterozygotes actually have an increased risk of developing a cancer because they already have one gene mutated, which means they only have to sustain one more mutation in order to get the disease. Okay, so we typically see in inherited cancer syndromes that it's a tumor suppressor gene. Now, the heterozygous cells do not form tumors because they need the second mutation, as I said. Heterozygous individuals will develop a disease just because they need a single cell out of millions to get another random mutation, just happens to be in the same gene. So it's a strong disposition to tumor formation that is inherited as an autosomal dominance trait, although it does have reduced penetrance, 90%. So if they don't get that random mutation, they lucked out and they don't get the cancer. Well, normal functions of tumor suppressor genes are to block cellular proliferation. So they participate in the pathways that regulate cell cycle. For example, retinoblastoma gene 1, this is the gene that's inactivated in retinoblastoma in the kids that have this. And this is a major regulator, as it turns out, in cell cycle control. It halts transcription of genes required for the cell to get into the synthesis phase. Again, it's a loss of function mutation. Sometimes what we see is, instead of a change in the nucleotide sequence, it is a result of the DNA becoming having methyl groups added to it, so it's permanently inactivated. And that's another way it, of course, will not be able to perform its function and induce an increased risk for tumor formation. Other tumor suppressor genes inactivate cyclin-dependent kinases, known as CDKs, again, that inactivate inhibitors. Okay. They again halt transcription, and sometimes they prevent cell-cell interactions. So they're, again, you're activating something that prevents, with the tumor suppressor gene, it prevents the cell from going through the cell cycle. And this gives us a picture of it, as you can see here. We're going to see the P53 and stuff in just a little bit, because this is very important in terms of DNA repair. But here are some of the others, and it's just showing you how it's related again to the cell cycle. Don't worry about knowing cyclin E and cyclin D1, etc., etc. Just get a feel for how these things are represented in different checkpoints. Let's continue using retinoblastoma as an example of an inherited cancer gene. So this was first identified in 1971 by Dr. Knudsen. I'm not sure we should just pronounce it Knudsen or Knudsen, it all depends. Anyway, it's a genetic form of retinoblastoma. Typically, there is an affected parent, and they have a 50% chance of transmitting it to each offspring. Unfortunately, it's usually bilateral. All cells have the inherited mutation, so it's a constitutional mutation. It just means it's there. But anyway, and the tumors then have a second mutation. And usually it turns out to be, as it says here, a large deletion of chromosome 13 that obviously is going to have to contain the RB1 gene. The bilateral tumors occur because you only need one hit, because they inherited the other. And the RB1 is a tumor suppressor gene, inhibiting tumor formation even when only one copy is present, as I explained in a previous slide. Now, in the sporadic form, neither parent is going to be affected, and it's usually just going to be unilateral, and that's because it will require two independent hits. He proved the study of inherited form by studying retinoblastoma helps identify genes important in the sporadic form. And that, of course, is also true for other genes, or other cancers, I should say, particularly breast cancer. So this figure shows you both inherited and sporadic forms of retinoblastoma. In the inherited, the sperm has it. So in the germline, so all the cells have it. Eventually a second mutation occurs in a somatic cell. Now you have cells that are homozygous for and the inactivated RB1 gene, and now they start proliferating. In the sporadic, okay, there are two ways. The first hit can occur in the embryo, or the first hit occurs in a somatic mutation, but not during development and everything. And then you got to get that second hit before you get a tumor. Here, if it occurs early on in the embryo, it can actually then go in sort of and occur at a similar age as an inherited, but this would be a situation where the parent is not affected. Which leads us finally to a discussion of DNA repair genes. And these are, if you have a mutation, I should say, in a DNA repair gene, what does that do? Well, it means that when the cell divides, and it makes a mistake, a DNA polymerase or something, a lot of times it can't be corrected. So we say that the genome is unstable, or genomic instability is the fancy term. And what happens is there's widespread mutations. You can even get chromosome breaks, and if this is occurring in, of course, the gametes, it can lead to aneuploidy. Now, there are a few rare inherited forms, but usually mutations in DNA repair genes are sporadic. Mutations, however, in BRCA1 and 2 and ATM, which are all DNA repair genes, lead to defective repair of double-stranded breaks in the DNA. And then you get faulty DNA mismatch repair. This can lead to colon cancer. And then one of my favorite ones to talk about, I just feel really bad for the people who have it though, is xeroderma pigmentosum. These are people who have a defective excision repair of pyrimidine dimers. Well, when they are exposed to UV light, this alters their DNA, and so they get these mutations. And they can't ever go out in the sun if they have the inherited form. They will get skin cancer. And so for kids who have this, they have camps where they only go out at night, which I thought was kind of nice to hear about. Just wish no kid had to have it. Anyway, this leads us to a very important discussion of telomeres. How does a cell know it's too old and it can no longer divide? Well, this is how normal cells become senescent after dividing a fixed number of times. Okay, how do they know to die out? What happens is every time a cell divides, the telomeres, which are just those repeats at the ends of the chromosome, begins to shorten. And it's just because of how the enzyme DNA polymerase works. We're not going to go into why. But so every time it will shorten a bit. When they get too short, the chromosomes start to fuse during mitosis and the cell dies. They can't separate. So we believe telomeres are the biological clock in cells, and they keep track of how many divisions that can occur. But hopefully you're thinking about, well, Dr. McDonald, what about stem cells, like the hematopoietic stem cell? Well, they keep dividing all the time. How come their telomeres don't get smaller? And that's because of an enzyme known as telomerase. And telomerase prevents the chromosome shortening and allows the cell to keep cycling. And not surprisingly, human tumors, 85 to 90% of them have an active telomerase in them. And this picture here just shows you the telomeres, and over here on the other side, why it gets shorter. I don't want you to worry about this, and I won't test you on it. The mechanism, just understand that the shortening of it is sort of like the biological clock of a cell. So how did we go about identifying inherited cancer genes? Well, obviously linkage mapping in families was the first way, and this had to be an informative meeting in order for us to get a handle on it, and those were kind of hard to get. Chromosomal deletions, of course, were easier to see, particularly if they're large deletions, and again, this typically would be a loss of a tumor suppressor gene. Remember, those are loss of function mutations. And if they were large deletions, they could identify chromosome region. Then you would try to do linkage analysis with markers and identify the loss of heterozygosity, because only mutated tumor suppressor allele present after the deletion. And we used this to identify the retinoblastoma gene and the Wilms tumor gene, and it took us a long time to find those. Translocations did speed up things a little bit, and that led to the discovery of MYC, with plasmacytomas or multiple myeloma, NF1, which is neurofibromatosis, and of course ABL, acute myelogenous leukemia, which we talked about with actually CML, chronic myelogenous leukemia. This shouldn't say acute, it should say CML. Oh well. And then, of course, the neurofibromatosis, the classic example is the elephant man, as you see over here. But I want to talk about DNA repair a little bit more and what they call the TP53. Now, this gene used to be called just little p53, okay? Now we're giving it TP53. I'm not sure why it's changed, but that's okay. Anyway, this is simply the most commonly altered human cancer gene around. More than 50% of all human tumors will have this inactivated. A result of chromosomal losses, deletions, but it's all on the short arm of chromosome 17. And we see this in colon cancer. Some of them show a missense mutation in the DNA binding region of the gene, so that can affect. But it's a tumor suppressor gene, but it also is very important DNA repair. Its expression is increased in response to the presence of double-stranded DNA breaks. Okay, so a transcription factor that controls the transcription of genes that allows cells to progress through a cell cycle. Okay, and if DNA is severely damaged, okay, the p53 will induce apoptosis. Now, when mutated, cells with damaged DNA may escape both repair and destruction because this TP53 is no longer working. So the cells continue to replicate, they can sustain more damage, and it can lead to tumor formation. Now, it turns out human papillomavirus, which is associated with the development of cervical cancer, will inactivate both the TP53 and the RB1 retinoblastoma gene. Some carcinogens seem to induce specific TP53 mutations. For example, aflatoxin B1 is produced by a fungus, usually in rice paddies and things like that, and it can interact with other things in the liver. Remember, it would be processed there in the liver. Benzopyrene in cigarette smoke, of course, and lung cancer seems to specifically inactivate TP53. There are some germline mutations in TP53 that leads then to an inherited cancer syndrome, Li-Fraumeni syndrome, LFS. This is going to be autosomal dominant, and they see all sorts of things, not just leukemias, but also carcinomas and sarcomas. And then this is one of those things where the presence of the TP53 indicates an aggressive breast or colon cancer, and the people have a poor survival. So research indicates that if we can insert a normal TP53 gene, it will lead to tumor aggression. So this is one of the gene therapy protocols that are in trial right now. We learned that tumor cells required more than one mutation because of studying colon cancer. Right now we know more about breast cancer than we do any other cancer, but colon cancer we understood better first, okay? And it was because of this inherited syndrome, which actually is a gene, familial adenomatous polyposis, or FAP gene. It's also known as adenomatous polyposis coli, or APC. Anyway, what happens in families that have the mutation in this gene is that they get these precursors, these polyps, colonic adenomas, prior to developing cancer, and they do however this is detected early and is removed, these little polyps, then it reduces the occurrence of colon cancer. Well, FAP is located on the long arm of chromosome 5, and it's a tumor suppressor gene, so both copies have to be inactivated in order for it to have an effect. but we identified it in families. But it turns out that it is also important in sporadic cases. So the same gene is also inactivated in about 85% of all the sporadic cases of colon cancer. So here was a situation where this became important in the majority of colon cancer cases. So early alteration in the progression from polyps to cancer is what happens when this particular gene is inactivated. Now, people who inherit the APC mutation develop, or FAP, however you want to think of it, again have a somatic loss of function mutation in the normal allele. Hundreds, not like the few polyps I had on my first colonoscopy, hundreds of these epithelial cells are affected. So you get lots of these polyps formed. And now some of the cases are not due to an actual mutation, but because the gene has not being produced because the DNA has been inactivated, too many methyl groups added to it. Other common mutations in colon cancer include a gain in function in a gene called KRAS, TP53 loss of function, another tumor suppressor gene, SMAD4. Appears that 7 mutations were required to get a colon tumor. And the reason we know that is because we would go in with these families early on and you'd take out the polyps, and maybe they wouldn't get all of the polyps, and then when they'd investigate looking at mutations, they said, very early on, the first one, say, was FAP. Then the next one was KRAS. And then actually we'll see on the next slide the actual sequence of the mutations. And it was all because we could see this in these unfortunate families who had the inherited syndrome. What does the APC protein do? It regulates another protein called beta-catenin. And this is an important molecule in the signal transduction pathway that activates MYC. And MYC, of course, is a transcription factor. And APC normally will reduce signals that lead to cellular proliferation. But when it's mutated, of course, it will not be able to do that. So APC mutations may also affect cell-to-cell and cell matrix adhesion properties. Again, it's mediated through controlling beta-catenin, and it's because of the role of beta-catenin in cell-cell and cell matrix adhesion. It interacts with E-cadherin. Well, don't worry about the E-cadherin and all this, but these things are on the surfaces of the cell that help them bind to other cells or to the extracellular matrix. So think about it, though. That's what keeps a cell in place. If they lose this, these adhesion molecules, they no longer stay in place. Now they're able to invade locally. So APC mutations may increase genomic instability, or additionally APC mutations may increase genomic instability by altering, again, chromosomal separation during mitosis. So all because of this mediation through beta-catenin, it can, if you lose it, you lose control of cellular proliferation, the cell doesn't stay in the right spot, and there may be abnormal chromosomal separation during mitosis. So it could really lead to other mutations, just with this one. So this shows you the progression of colon cancer in these families. So we start out with the loss of APC. Then other parts of the DNA gets hypomethylated, so it gets active. Here's the KRAS is the next one. Then SMAD4. So we got activation of a proto-oncogene, so now it's an oncogene. Another loss, and here's the TP53. And then finally we have to have other alterations before they can metastasize. Now it turns out there are other colon cancer genes, the one called hereditary non-polyposis colon cancer genes, or HNPCC. I'm not sure which is easier or harder to say. It's up to 1 to 5% of all cases of colon cancer. Now, this one's an autosomal dominant, so it has a 70 to 90% risk in heterozygotes. It also has a high risk for endometrial cancers. And mutations in 4 different genes account for most of these, so they all play a role basically in poor DNA repair. And so this increases the rate of mutation. This particular repair is called mismatch repair. And if the genes for mismatch repair aren't working properly, then the DNA mutations that occur during normal cycling don't get fixed, and they may, of course, inactivate a tumor suppressor or activate a proto-oncogene. They can also lead to microsatellites that become unstable. And there are a lot fewer polyps, hence the non-polyposis, than in FAP, but because of a high rate of mutation, they have a higher risk for tumor development. Well, we can't talk about cancer genes without talking about breast cancer because of its high prevalence, lifetime prevalence in women. When I started talking and learning about all of this, they said it was 1 in 9 or 10. Now it's 1 in 8, and it doubles if you have a first-degree relative that had breast cancer. 1 to 3% of cases are due to inherited mutations in BRCA1 and BRCA2, and this increases your risk, of course, to 50 to 80%. Mutations more common among women with early onset, positive family history of breast cancer and ovarian cancer. So the BRCA1 mutation increases also the risk for ovarian, prostate, and colon cancers, whereas the BRCA2 increased risk of breast and ovarian in women and breast cancer in men. And surprisingly, I've known a couple of gentlemen who have developed breast cancer. Well, how did we identify all of these genes? Actually, by linkage analysis and then positional cloning. Both genes are large, and so lots of different types of mutations have been identified for BRCA1 and BRCA2. But the loss of function, complete loss of function, requires, again, 2 hits because it's a tumor suppressor gene. We, however, do not see mutations in BRCA1 and 2 very often in sporadic cases. So this is one of those things where, yes, it's true for the inherited form, but not necessarily for the sporadic forms. However, both participate in DNA repair again of double-stranded breaks. And you may have noticed that I've called DNA repair sort of tumor suppressor, and they are kind of similar. So they're both a loss of function. And that's why traditionally DNA repair were considered under the tumor suppressor, and they've just now been separated. And so sometimes I have a tendency to call one or the other the same. So I try to get better at that. But anyway, BRCA1 and BRCA2 are important double-stranded breaks in DNA, and when they get inactivated, again, you have incorrect repair and the genome is unstable. Now, we also know that BRCA1 and 2 interact with other tumor suppressors, such as TP53, RB1, and MYC. Other inherited mutations can lead to breast cancer. So here is the mechanism for repair of double-stranded breaks in DNA. And you may have heard of the ATM gene. It is not, of course, where you can get some money. But anyway, I couldn't resist. Sorry about that. So BRCA1, typically we get phosphorylated in the presence of double-stranded breaks, and then they would go and help with the RAD51 to repair the breaks. And if they can't do that, then the breaks stay, and you may have inactivated a tumor suppressor. There are more inherited cancer genes. Familial type of melanoma, again, it's a tumor suppressor. Multiple endocrine neoplasia type 2. This is a RET proto-oncogene, a gain of function in this one. Hirschsprung disease, a loss of function mutation. And many more. There's been about 30 more, and of course you're not going to have to know all of them. But the importance of inherited mutations in common cancers, well, The presence of one affected first-degree relative usually doubles one risk. Okay. Additional genes are shared within different families. Families, however, also share environmental factors, so sometimes it can be hard to determine what's really important. So identification of cancer predisposing alleles, we have to look at all the genes involved in a particular cell pathway to find the one that really may have been inactivated. And if it's all in the same pathway, several different genes and different mutations can lead to the same cancer phenotype. So we need genome-wide association studies, animal models, then we look for the altered genes in tumors, and this all leads to new diagnostic and therapeutic tools. So we'll be talking about these diagnostic and therapeutic tools as we go through the last chapters in the textbook, which are more of cases and taking all this fundamental background information and applying it to a real world situation. Otherwise, after listening to this and doing some study, you are ready for that first exam. So I will say goodbye to you today with this, and I will be speaking with you some more next week. Good luck on the exam.